Hello students, in the last video we studied enzymes and then the last topic was industrial application of enzyme catalysis. Now next topic is nucleic acids. Nucleic acids, you studied a lot of syllabus or lot of portion of this nucleic acids in biology. Now here with the chemistry point of view we are going to study nucleic acids. Okay, so what is this nucleic acids? Nucleic acids are one of the most remarkable properties of the living cells and their ability to produce their replicas through thousands and thousands of generations. Through thousands and thousands of generations. In Marathi we always say like that, बाळ जन्माला आलं की अरे हां हे आईसारखं दिसतं आहे, नाही 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 वडिलांसारखं दिसतं आहे, नाही नाही हे तर आजीसारखंच दिसतं आहे. हां तर व्हॉट इज दॅट? That is nothing but replicas through thousands and thousands of generations, and those are transferred by these nucleic acids. All these properties, remarkable properties, are produced due to these nucleic acids. Okay, and here the certain type of information is passed and that is also unchanged from one generation to the next. This is very important. This information is passed from one generation to the next and that is also unchanged. Okay, and this genetic information transferred through these nucleic acids, These nucleic acids are two types. One is RNA, another one is DNA. What is RNA? RNA is ribonucleic acid while DNA is deoxyribonucleic acids. RNA ribonucleic acid, R stands for ribo, N stands for nucleic, A stands for acid. Here D, we are not mentioning here R, we just mentioning here like this, D for all deoxyribo, then N stands for nucleic and A stands for acids. Okay, so this is DNA, this is RNA, ribonucleic acid and deoxyribonucleic acid. Deoxy means OH is less as compared to this. So this is deoxyribonucleic acid. What is this? Here see. First of all we have to see the structure of nucleic acids, and this is the main aspect of our this chapter. We have to study the structure of nucleic acids. Okay, nucleic acids are unbranched polymers of repeating monomer called nucleotide. We have to see what is nucleotides. Okay, means this is nothing but polynucleotide structure. Nucleic acids are unbranched polymers, means so many polymers, means so many repeating monomers. Monomers, which monomers? Monomers which called nucleotide. We have to see this, what is nucleotide. When it is polymer, means this is polynucleotide structure. Okay, this DNA is present in the nuclei of living cells, while RNA is in the fluid of living cell. Okay, you studied this in the biology. DNA present in the nuclei, and while the RNA present in the fluid of living cell. In this DNA there are several million nucleotides. Million means 10 lakhs, several million nucleotides, while RNA is having few thousand of nucleotides. This is the difference in between these two. Okay, but both are the very important remarkable properties having with these parts of our living cells. Okay, now next, these nucleic acids made up of three major components. Okay, means in a nucleotide monomer there are three major components. Which are these three major components? First one is the monosaccharide, second one is nitrogen containing base. These are 5 structures are there, and the third part is the phosphate group. Means this nucleotide monomer is made up of 3 components. First one is monosaccharide, which we studied very well, then nitrogen containing bases, and then the last one is the phosphate group. We will see one by one. In RNA made up of D-ribose, D-ribose is nothing but furanose. What is furanose? We studied in glucose. Furanose is 5-membered furan like structures, that's why we called it furanose. Then we studied fructofuranose, that is nothing but derivative of fructose, like the fructofuranose. Okay, but now here it is D-ribose, and this D-ribose structure is like this. In our glucose, here again at this position there is CH2OH like this, but when we are studying D-ribose, here it is just OH. This is not present over there like glucose, like derivatives of the glucose, whatever there. Okay, so here it is just OH and the first carbon, not CH2OH, and H is there. Then now this structure is known as D-ribose. This structure is known as D-ribose. See, 1, 2, 3, 4, 5-member ring, just like a furan. Okay, then when we are going to numbering, this is first carbon, second carbon, third carbon, fourth carbon, and fifth carbon. Here it is the OH to this side and H to this side. All these anomeric carbons related theory we studied in first topic of this chapter. Okay, this is there. Then in a DNA, monosaccharide is 2-deoxy-D-ribose. At the second position, this is the first carbon, this is the second carbon. Deoxy means this no OH. See, here it is at the second carbon there is OH, but at the second carbon there is H. No OH at C2. That's why this is known as deoxy. Please try to understand what is D-ribose and what is 2-deoxyribose. Ribose is this structure. Okay, this is again the same ribose, only at the second carbon, there is no OH. No OH. See here, this is the second carbon, there is no OH, there is H. Here it is OH, here it is H only, no OH. That's why this is deoxy. Okay, then another important component of this nucleotide is nitrogen containing base. These are derived from two types of parent compounds. Which are two parent compounds? One is purine. When there is a 6-member ring, here it is nitrogen, here it is nitrogen. Okay, this is the parent compound purine. Now these are the derivatives of the purine. First one is a thymine. Okay, so what is the difference in between these two? See now here at this position there is C double bond O, here it is C double bond O, and at this carbon there is CH3. Then this is known as thymine, and short form is T. Thymine, short form is T, and this is present only in DNA. Again remember this thing, this thymine T is only present in DNA. Next one is uracil. What is the change? See here it is at this position C double bond O, at this position C double bond O, remaining things are as it is. This is known as uracil, short form is U, and this is only present in RNA. One more base is there, that is a cytosine, short form is C. See now, this is the parent compound purine. Here it is at this carbon, here it is C double bond O, then at this carbon here it is NH2. This is cytosine. So you can remember all bases, thymine, uracil, cytosine, by knowing the parent compound and what are the derivatives of it. You have to remember all these additional groups attached to this, or the presence of this C double bond O at this carbon, or to this carbon CH3 is attached, or to this carbon NH2 is attached. You have to remember these things. Okay, so this is thymine, uracil, or cytosine. Then next parent compound is sorry, I did some mistake, I think. Sorry students, this is our pyrimidine, okay, and this is our purine. This is pyrimidine and this is our purine. Okay, now what is this purine? Purine is now, this is a 6-member ring, and to that there is a 5-member ring is attached. This is pyrimidine, while this one is purine. Okay, this is parent compound. And now to this compound, at this carbon here it is NH2 attached. Then in the next adenine there is the NH2 attached, it is known as A. While in guanine here it is C double bond O, and to this carbon here it is NH2. This carbon, this C double bond O, and at this carbon this is NH2. So this is adenine A and guanine G. Short form is G. And one more thing, that is the phosphate group. What is this phosphate group? We are going to see now. Okay. So these are the three major components. Then what is nucleoside and nucleotide? First of all we have to know, and then we will go to the whole structure. We will go to the whole structure of this our monomer. Okay. See, what is nucleoside? It is formed by joining the anomeric carbon of the furanose with nitrogen of a base. We studied those 5 structures of bases, and these bases structure as added to or the joined to furanose. Okay. How? When there is a pyrimidine base, like this, okay, then the nitrogen atom at the one position of the sugar it is attached. At the one position of the sugar it is attached. While in purine bases, the nitrogen atom at the 9th position with first carbon of the sugar, that is the C1 position. This is our C1 position. See, this is our C1 position. This is 2, this is 3, this is 4, and this is 5. This is our C1 position. Okay. So they form nucleoside. This structure means monosaccharide and base, they gives you nucleoside. And to this nucleoside, when there is a phosphate group attached, then there is formation of nucleotide. Please try to understand. First monosaccharide, then the base, it gives you nucleoside. With this structure, if the phosphate group is attached, this is the third component, then it becomes nucleotide. Nucleoside, two things, sugar and base, monosaccharide and base, nucleoside, monosaccharide, base and phosphate, it becomes nucleotide. Okay. And how these are going to form? It is formed by adding phosphate group to the 5th OH of the nucleoside. 5th OH. Which one is the 5th OH? This one is the 5th OH. Okay. And thus nucleotides are monophosphates of nucleosides. This is nucleoside and one phosphate group attached over here. So this is monophosphate of nucleoside. And short form is MP. MP, monophosphate, DP, diphosphate, TP, triphosphate. This is monophosphate. Now see here. We know the fructofuranose, at the C1 position there is CH2OH. But now here it is just OH, this is our D-ribose. Okay. Now adenine, this is the base, which is the parent compound is the purine. Okay. And here this is attached to the C1. This is attached to the C1 position. So this is attached to this C1 position. See here. This is attached to the C1 position. Then this OH and H from this here is going to remove, and this adenine ribonucleoside is formed. This OH and H from this H and this OH minus H2O, and this adenine ribonucleoside is formed. And now the third component is going to attach, that is the phosphate group. P double bond O minus, this is PO4 phosphate group. We know this very well, phosphate group. Okay. When it is going to attach, it is going to attach to the 5th, this adding phosphate group to the 5th OH of the nucleoside. Here it is. This is the position, fifth one, this is the fifth position to this nucleoside. So this phosphate group is attached over here to this position. See now, and this thing is going to happen. Okay, and here it is formation of CH2O, P double bond O, OH, OH. You may write here O minus also, that doesn't make any difference, or you may write OH, OH. But writing a charge means is always a very reactive, but to get a stable whatever the product, we should write here OH, OH. And this is the reaction condition also. So in the textbook it is given O minus, but for the understanding I am writing here OH, OH. Okay, what is this? This is nothing but adenine. This is our adenine. Then this is only one phosphate group, so this is monophosphate. Then here it is the ribose, so this is ribose, and this is nucleotide. But in the short form we just write adenine monophosphate, because this base is going to remain. When we are going to use D-ribose, this base is going to remain same. Only here it is adenine, you may use adenine, or you may use guanine, or you may use cytosine, or you may use uracil, or you may use thymine. You may use means it may be present over there. Okay, so different combinations, different nucleotides we are going to get. Okay, this is our AMP. In our textbook, just example is given with the MP with the product only like this. While formation of nucleoside, they use different bases. All these things are here, that's why I took whatever the product here to understand well. Okay, not just like a textbook, I take different ways to understand what is AMP and then what is DCMP. Okay, so we are going to now see the DCMP. Here it is, DCMP is made up of 2-deoxyribose, and this C stands for cytosine. This is the structure of cytosine. The parent compound is pyrimidine. This is parent compound pyrimidine, and this is our cytosine. NH2 is here, and C double bond is here. These two are going to attach to here at the C1 position. This is 1, 2, 3, 4, 5. Okay, this is the second position. There is no OH. That's why this is 2-deoxy. Okay, here it is, this is attached. So at this position, and this nitrogen is going to attach to each other like this. NH2, 6-member ring, N and C double bond O, and this is our furanones, that is deoxyribose. Now one phosphate group is going to attach over here, and we know that phosphate group is attached to here at the fifth, 1, 2, 3, 4, 5, at the fifth position OH. So here at the fifth position of the OH, phosphate group is attached. This is only one phosphate group, so again this is monophosphate, MP, monophosphate. Okay, now what is this compound C? This is cytosine, and this is deoxyribose. When these two are attached to each other, and form this type of product, then this is nucleoside. What is the name of this? This is cytosine deoxyribose nucleoside. Deoxyribose nucleoside. When phosphate group is attached to here, then this become monophosphate. Then this is cytosine monophosphate deoxyribonucleotide. But when we are writing short form, for deoxyribonucleotide we writing only D, then for cytosine C, and for the monophosphate this is MP. Try to understand why the name of this nucleotide is DCMP. Means what? D means deoxyribo. This is deoxyribose. C for cytosine. MP for monophosphate. So this is DCMP. Okay, and now We will go in more details the structure of nucleic acids in the next video. Thank you very much.